Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 171st seminar, and I call it Lawrence Berkeley National Lab Day. Since August 26, 2021, when the first SynBYSS webinar started with Professor Francis Arnold, a Nobel laureate, Teresa Good, the NSF Bio MCB director, India, the EBRC director, and Doug, the Biomed CEO, and Albert, a now tenured professor at NC State University. The SynBYSS speaker community has grown tremendously with a total of 450 speakers and more than 2,000 audience. The pioneer speakers include a Nobel laureate, 26 National Academy members, including today's speaker, and 44 program directors of almost all US funding agency and some EU funding agency, and 11 editors of Nature and Cell journals, and also 20 editors in chief. We have now 212 rising star speakers, many of whom have received tenure or secured a faculty position. Notably, one eligibility criterion for rising star is that they should be an untenured professor or rank below, and many of them a few years later actually become a tenured professor. My YouTube channel has also attracted more than 2 million views combined, reaching out to the global community. Celebrating this passionate community members' collective success to form a ever-increasing community, so much all SynBYSS people for your enthusiastic support and participation, and I will continue to build a diverse and collaborative community with you. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Blake Simmons. He is a division director at Lawrence Berkeley National Lab and vice president of Deconstruction Division at Joint Bioenergy Institute, also short JBA. He was also elected as 2024 Fellow of the National Academy of Inventors, one of the highest honor in science and engineering. He is a true pioneer with expertise in biomass deconstruction. That is a very important area for making a good or great bioeconomy. I have known him for many years, and I thank him for accepting my invite last year when I met him at a conference again. Blake, thank you so much again for your amazing contribution to the research community, enabling biomanufacturing and bioeconomy, and the virtual podium is all yours now, and thank you again.
Thank you very much. It's an honor to be here. And, taking back at the accomplishments that you've been able to achieve over 4 years are really remarkable. So thank you for everything you've done for the community and for outreach to science writ large. You are a true ambassador, unparalleled in the community. So thank you again for everything that you do. So with that, I'm going to turn it over to science now and give you a little discussion and talk about some of the work that we've been doing in the bioeconomy writ large. This is a really exciting project on the demonstration and scale up of an integrated feedstock flexible technology to make a synthetic aviation fuel 1,4-dimethylcyclooctane. The vision here is that if the future is going to be fermented, it needs to be built on cheap sugars, meaning you have to unlock the potential of lignocellulosic biomass to generate sugars that are cheaper than conventional sugars on the world trade market. And so you need to be able to access a diverse range of feedstocks. You can't just be limited to one or a very narrow range. If you really want to move the needle in the bioeconomy, it needs to be distributed and worldwide. And biomass is distributed and it is varied and it is different everywhere you go. And so we want to be able to take these feedstocks or mixtures thereof and break them down into fermentable sugars, convert those sugars into something like isoprenyl acetate, shown here. And then we're bringing a chemical catalysis step to cyclize this compound and form 1,4-dimethyl-cyclooctane. We'll call it DMCO from here on out, just for the sake of brevity. We've got a really great team. It spans Berkeley Lab, University of Hawaii Hilo, the China Lake Naval Air Warfare Station, and the Washington State University. What we're trying to do is work with strains like Pseudomonas petita and E. coli. This is all about hydrolysate conversion. So this is nothing being done in mock hydrolysates or defined media. We have a team working on the biomass deconstruction. We have a really great microbe team led by Tixon Lee that's engineering these strains to produce isoprenyl acetate. We have a scale-up facility here at Berkeley Lab called the Advanced Biofuels and Bioproducts Process Development Unit that is scaling it up from the bench and taking it up to 300 liters of fermentation. And then we're handing the isoprenyl acetate off to the team at the Navy to form this DMCO. Our key performance metrics are greater than 90% yields of fermentable sugars. We want to go to greater than 18 grams per liter IPA production on a hydrolysate, which is to be the first ever demo of its kind, and greater than 98% DMCO catalytic yield, which the Navy team has already achieved. So why DMCO? What's so special about it? Well, it's a compelling aviation feedstock because it is a higher energy density than Jet A. It means a plane can go further on the same kilos of fuel. It's also cleaner burning. It reduces contrails, and it can replace aromatics that are required to maintain seal integrity. That is really important to enabling 100% SAF flight. And so we're really excited about this compound. We're in the process of working with industry to take it to market. But we're going to focus right now on some of the work that we're doing in the project. So we've really focused on dimethylamine as a really innovative solvent that is very good at breaking down biomass. It is very volatile, meaning that you can pre-treat the biomass and then pull it out and recover and recycle it very effectively. We get above 99% recycle and recovery rates typically in the lab and at scale here at the AVPDU. When you get the pre-treated biomass, you then add enzymes to liberate the sugars from it, and then you get a sugar-rich hydrolysate. So a very simple process. Not a lot of intervention here. And we've actually gone to the step where we can integrate the pre-treatment and the enzymatic hydrolysis in the same tank, and then just deliver the hydrolysate as a liquid to the fermenter to then be converted into isoprenyl acetate. When we screen the DMA conditions, just to show you how effective it is, this is 40% dimethylamine in water, 22.5% dimethylamine in water, and 5%. We're screening from 40 to 120 degrees C. These are the sugar yields over here on the left. And you can see we're getting 90% yields of glucose and xylose pretty consistently until you get down to 5% and lower temperatures. What is remarkable is that at 40 degrees C, you're operating as well as you get in 120 degrees C, which challenges the conventional wisdom about what makes pretreatment work, because the notional wisdom is that you need to get above the glass transition temperature of lignin, which is above 120 degrees C, in order to have an effective pretreatment. So really exciting stuff, and DMA outperforms pretty much any other solvent that we've worked with at a variety of conditions. Just to give you an idea of what it looks like as you operate with it, this is when you're drying the biomass after pretreatment. This is in a 50-liter tank in the early drying stage. As you get to more drying out, it becomes a wet solid, and then when you get about 99%, it is basically a dry powder phase. So again, just showing you the process that is very effective to remove the solvent. We've also demonstrated that we can recycle it 3 times with no loss of performance, and we don't think there's any limit to that. We think we can recycle it 100 or 500 times without any losses in terms of sugar yield. Here's the enzymatic hydrolysis process. This is when we start, when we just add the enzymes. You can see it's looking a lot like saltwater taffy. 30 minutes in enzymatic hydrolysis, it really turns into a viscous liquid. But within 24 to 48 hours, this is looking like a great hydrolysate that's suitable for fermentation. Okay, come on. Got to go on. So this is what where the exciting stuff happens in terms of integrating with the synthetic biology. Here's an engineered host that we feed on the hydrolysate. We also fed it a mock. What is interesting to note is that the hydrolysate outperforms the mock hydrolysate, even though those are the same glucose and xylose yields. We think there's some micronutrients that come with the hydrolysate that actually boost performance. We're getting close to 11 grams per liter. And this was a couple weeks ago. We're now up to 15 grams per liter, and we're trying to push it above 20. You can see where the headroom is. We're getting really good glucose consumption, but we're only getting about 40% of the xylose consumption. And so we've tightened up that pathway for the co-utilization of glucose and xylose, and that's how we're going to be achieving some higher titers. We've also seen an intermediate in this pathway, mevalonate, is being produced at significant levels and not converted. So we have a bottleneck where that metabolite pool is accumulating intracellularly. And so we've also tightened up that pathway to really streamline the carbon conversion efficiency. So just to wrap up here real quick, we're going to be continuing the E. coli strain engineering. We're going to optimize the xylose utilization pathway. We're also going to be pivoting to a high gravity fed batch at 50-liter scale and then going up to 150. And then we're going to be on track to do the 300-liter campaign run in September and October of this year. The techno-economics of this are really exciting. Just to put it here, this is the current state of technology. This dotted line is a commercial price of dextrose, the wholesale price. You can see we're already beating that, which is pretty cool. And we think we can actually get to lower than 50% of that dextrose price if we meet all of these different process improvements. And these are not a Herculean lift. So we're looking forward into the next 2 years. We'll be scaling up this process even further. We'll be working with industry to do that. And we're also looking at the potential of co-cultures to really come up with a SynCom approach to boosting overall productivity and also converting some of the lignin-derived intermediates that we can liberate. So here's the team, very wide-spanning team from across the world. And with that, I thank you for your time and turn it back to you.
Thank you so much. This is amazing. I mean, synthetic biology, I would say, started 25 years ago. This is time to deliver real product, not just working on the toy system. So this is really amazing. Also, I'm glad because you are working on the, aviation fuel, because even if we have a lot of electrical, energy, battery, we don't want to fly to Hawaii using the battery because that's not possible at this moment.
Yeah, I always tell people I'm not going to get on a plane that's powered with an extension cord. That's not, a viable path forward currently. So I completely agree with you on that front, my friend.
Absolutely. Thank you so much. This is amazing. All right. Let's go to the next speaker. Okay, now the main speaker of today with a longer introduction. Dr. Juhyun Kim is a professor at Kyungpook National University, Korea. Juhyun's research focuses on bacterial gene expression, which has naturally extended his focus to both molecular and synthetic biology. A key aspect of his work involves investigating the intrinsic constraints that affect gene expression. His research explores not only how cellular resources are allocated to heterologous genes under dynamic growth conditions, but also how to enhance the activity of such genetic circuits in bacterial systems. In addition to his expertise in synthetic biology, he has honed his skills as a molecular biologist. As a graduate student, his passion for Pseudomonas species led him to Spain, where he worked with Victor del Loranjo, another pioneer speaker of this seminar series, to study the soil bacteria. Among his projects, he mapped the transcriptional profiles of toluene biodegradation using RNA sequencing and tilling arrays. He also identified the structural framework of the gene expression machinery responsible for the toll catabolic system in Pseudomonas putida. Juhyun, thanks so much for your time today, especially joining us from very inconvenient time zone, I mean past midnight. Please take it away, all yours now. Thank you again.
Well,
Thank you. Thank you so much for your nice introduction. Let me share my slide. Okay. But now I guess you can see my slide, right?
Yes.
So, again, thank you so much for the introduction. And I'd like to say hi again if someone is here. Well, just in case, my name is Juhyun, also known as Ju, as my international colleague call me Ju. And I'm working at Kyungpook National University, and it's great pleasure to be here with you virtually and to present and share my work. Today, I'm going to talk about programming bacteria by manipulating resource allocation. I guess, as many of you know, and as already Blake told us, bacteria can be programmed to carry out specific functions, including well, therapeutics, bioremediation, product fermentation, and etc., etc. This process involves designing and introducing genetic circuit into bacterial cells to enable them to perform such functions. However, programming bacteria is far more complicated and complex than programming computers or electronic devices, as the implementation often poses significant challenges. For instance, some genetic circuits may not be stably maintained or function properly within the cell. One of the main reasons I think is that we do not fully understand the gene expression hardware of the host cell. Specifically, we don't know how the cell allocates its or their limited cellular resources across different functions and genes. Speaking of this, let me show you an example of bacterial programming to control pathogens. In one study, researchers successfully engineered E. coli to target and kill Pseudomonas aeruginosa. They basically designed and built a genetic circuit with a quorum sensing based regulator and a specific bacteriocin. So when the population of uh, Pseudomonas aeruginosa grows and releases its signaling molecules, the engineered E. coli detects them as a autoinducer and attack the pathogen by producing the bacteriocin. When tested, as you can see here, the system functioned effectively as the evidenced by the clear halo formed through the lysis of Pseudomonas aeruginosa. To further improve the lysis effect, the researchers added antibiofilm coding gene into the circuit. But interestingly, the lysis rate did not increase at all. They found that for this problem is that when both genes were expressed at the same time, the level of bacteriocin went down. This happened because of the gene expression coupling. The gene expression coupling means that when two or more than genes are expressed simultaneously, even if they are not directly linked to each other, the expression of one gene can be affected by the other. That's because the cell has only limited cellular resources, including RNA polymerase and ribosomes. Well, just for helping you understand, let's think of it this way. Let's say you have $50 each to spend. With that budget, you can buy either 25 Cokes or 6 pizzas if you spend it all only for just one item. Of course, you can also mix them. For instance, you may get one pizza and 21 drinks. Alternatively, you can buy 5 pizzas and 5 drinks. The most important thing is that every time you choose one pizza, in this case, you need to give up the chance to buy poor drinks. This trade-off is exactly the same thing we already observed in the study with the gene expression coupling, and as a result of that, the programming with the dual toxin expression system did not work effectively. By the same token, when cells put more resources into one gene, they automatically have fewer resources. I mean, bacteria itself have fewer resources left for another or the other genes. Well, to characterize and measure the gene expression coupling level, we can use this simple genetic circuit. Basically, it has a constitutively expressed GFP reporter along with an inducible RSC expression system by AHL. When tested, as the cell produced more RFP, the level of GFP decreased again due to the gene expression coupling. In other words, there is a trade-off. To gain 1 unit of RFP, the cell needs to give up about 1 unit of GFP. It is thus necessary this gene expression coupling for effective bacteria programming. Actually, it is known that this gene expression coupling is mainly caused by limited translational resources, such as ribosome, because when the RBS, I mean ribosome binding site strength for RFP was lowered, the coupling effect disappeared. Based on this, and given the information to break the gene expression coupling, we can separate ribosome pool. And that would be like getting a pre-coupon per pizza, as I already had a chat with you for the example. With that coupon, you don't have to give up your drink to enjoy a pizza. In the same way, if the cell has access to a separate pool of ribosomes, the gene expression coupling would be gone. To do this, we split the ribosome pools by introducing orthogonal translation system into the bacterial cell. Well, this orthogonal translation system is not bring from other organisms like T7 RNA polymerase, but this system is created by modifying the Shine-Dalgano sequence, and it's complementary anti-Shine-Dalgano sequence in the 16S rRNA. Once the orthogonal ribosome is generated, it does not recognize the host RBS, but instead specifically interact with the orthogonal ribosome binding site. Using a well-characterized orthogonal ribosome binding site previously identified and its counterpart 16S rRNA sequence, I redesigned the genetic circuits so that each reporter can be translated by a different ribosome, and we tested how this affected the level of coupling. So when both genes were translated by the native ribosomes, as we already had a chat, we observed clear gene expression coupling. In this case, for every unit of RFP produced, the cell had to give up about 3 units of GFP. Similarly, when both genes were translated by orthogonal ribosomes, the coupling was still there because the two genes were now competing for the limited pool of orthogonal ribosomes. On the other hand, when two genes were translated by a different ribosome pool as orthogonal ribosome and host native ribosome, we still observed some coupling, but it was very, very minimal. With this engineering and with this separation, the cell only had to give up about 0.07 units of GFP to make 1 unit of RFP. Accordingly, the gene expression coupling is attenuated by separating the ribosome pool per each gene. Building on this idea, I constructed a DNA circuit for biolysine biosynthesis. The pathway requires the BOABCDE genes, and I separated their expression as BOA was placed under the orthogonal ribosome system, while BOBCDE were grouped together and translated using the host ribosome. With this setup, the cell produced more biolysine than controls synthesizing all the biolysine associated protein with the host ribosome, because the genes do not have to compete for the same limited pool of ribosomes. Well, another way to break this coupling is to increase the number of ribosomes available in the cell. Think of it again like this. If you are a billionaire, you don't have to give up 4 drinks just to get 1 pizza. You have, because, enough money for both, and you can buy them as many as you want. Well, it is known that bacterial cells carry more ribosomes when they grow fast. Based on this, one simple way is to make the cell one simple way to make the cell billionaire or rich is to grow bacteria under fast growth condition, which produce lots of ribosomes in the cell. To do that, and to study the relationship between cellular growth and the gene expression coupling, I built a homemade chemostat, which you can see in this slide. Of course, this is not the same level as fully automated liquid handling system, and as Blake showed very briefly, but at least it gave us precise control over cellular growth by controlling dilution rate. With this setup, we confirmed that as the growth rate increased, the key cellular components such as DNA and ribosomes also increased according with previous reports. But very surprisingly, the level of coupling did not decrease at all regardless of growth rate. Even we expected if they had more ribosomes, probably the coupling level would be released or attenuated. But actually, when the cells grew faster, the reported activity was even lower. Please have a look at this relation, growth rate and the gene expression level. Again, if cells grow faster, unfortunately or unexpectedly, the gene expression level of the GFP or RFP was lower. Later, we learned that using computer simulation and several experiments, when cells grow quickly, they tend to allocate most of their resources to ribosomes rather than other cellular fractions. But under nutrient-poor conditions, they divert more ribosomes to non-essential genes, including circuit genes, at the expense of cellular fitness. This cellular economic behavior eventually can shape the functional system we want to implement, and we observed the same outcome when programming bacteria with the type 3 secretion system. Type 3 secretion system is found in many gram-negative bacteria. They use it to invade host cell by delivering effector proteins directly into them, and it helps to invade mammalian cell. Well, apart from that, type 3 secretion system has been used to secrete usable proteins, including spider circle protein without complicated purification steps. Also, using this secretion system, recently therapeutic proteins can be delivered into cancer cells. We also built an IPTG-inducible type 3 secretion system and tested its functionality in Salmonella. By tagging the basal body of the secretion system with GFP, we observed that most of the signals were localized at cell periphery, which means that the functional proteins are synthesized. But interesting is that when the system was strongly activated while growing the cell in rich medium as LB, the reporter activity actually decreased. To test its functional capacity, we also introduced an expression system of type 3 secretion system dependent effector. When we measured the secreted output, we found that no effector was detected in cultures only when high level of inducer as 200 micromolar of IPTG treated. Well, this result is not due to some unknown regulatory network in Salmonella because we observed the same phenomenon when we tested the system in E. coli. Well, next, we also fine-tuned the expression level using CRISPR inhibition system, and the suppression effect was still present. Also, RNA sequencing analysis showed that transcription of the secretion system increased linearly with IPTG concentration. These results tell us that the suppression was not due to the transcriptional burden, but rather translational burden. We also found that ribosomal genes were downregulated when the system was strongly activated, which means that pure ribosomes could be accumulated in the cell. In this culture condition, in rich medium, cells prefer to maintain their own cellular fitness. Again, they prefer to invest ribosomes to make ribosomes, as I mentioned earlier. As a result of that, pure ribosomes are available to be allocated for the secretion system. On the other hand, when the cell, again carrying the secretion system, were grown in minimal medium, the suppression was not observed. Again, based on their cellular or economic strategies, bacterial economic strategies, the cells invest pure ribosome in growth under these conditions, in slowly growing condition, and this allows them to allocate more ribosomes to the secretion system, and it can be helpful for effective cellular programming. So collectively, what we learned is that effective bacterial programming is not about making them rich, but about spending their resources optimally, balancing growth with the functional system. Well, one way to achieve this could be through feedback controller system to control and regulate growth, or by using host-independent translation system like orthogonal ribosomes. Well, today I did not bring these approaches, but I would like to share the result with you in the near future. But instead, I'd like to share with you an extra gear for controlling resource allocation. In addition to cellular economic behavior, we also need to consider the physical constraint of the gene expression hardware. A growing number of observations showed cellular components in bacteria are spatially organized within the cell, although they do not have intracellular membranes. For instance, ribosomes are not randomly distributed in E. coli, also in Bacillus. While they are often found in specific regions, ribosomes are found in specific regions, especially near the cell periphery. This is because chromosomal DNA is highly compacted in the center of the cell, and the large molecules like ribosomes cannot occupy the same space, so they are effectively pushed outward. Not only these cells, when I labeled RNA polymerase with GFP and ribosomes with RFP in Pseudomonas putida, as you can see on this slide, the two molecules were clearly segregated. Again, RNA polymerase was mainly localized in the central region of the cell, while ribosomes were concentrated toward outside. This suggests that most bacteria have a similar structure in their gene expression hardware. Considering this gene expression structure, when I visualized the specific transcripts from the toll and catabolic system, I found that it was not located on the template DNA. Instead, it accumulated in ribosome-rich regions of the cell. Again, this means that RNA molecules can have ability to move around the cell. This result also suggests that not all genes are subject to transcriptional and translational coupling, as transcriptional and translational machinery is physically compartmented, and some mRNAs can actually relocate to ribosome-rich regions after transcription. While taking into account cellular structure and molecular dynamics, we can also engineer bacteria-derived mini cells for effective programming. Bacteria mini cells are small, unnucleated cells produced by abnormal cell division. Since it does not have chromosomal DNA, it cannot be reproducible, and owing to this property, we can use these mini cells for diverse applications, including drug delivery platform. While to produce mini cells, we can disrupt the cellular division system. Basically, bacteria can be divided symmetrically with the help of proteins such as MinZ, D, E, and FDSZ. The FDSZ ring is the key protein that drives division at the center of the cell. To prevent FDSZ ring formation at periphery or polar regions, the MinZ-D complex plays a role to degrade any FDSZ ring that form in such regions, if the MinZ genes are inactivated, the cell can divide asymmetrically, and as a result of that, mini cells can be produced. Once mini cells are produced, they can be isolated using differential centrifugation, and the residual parent cell can be removed by filtration and antibiotics to inhibit further cellular growth. But to use mini cells for a particular application and for effective programming, we need to program the mini cell producing strain. However, as I already showed you, not all the components we want will accumulate in mini cell because of the cellular structure. We also confirmed that using comparative proteomics between parent cell and the mini cell, and we found that, for instance, DNA-associated proteins were more enriched in parent cell, while proteins related to transport and cell cycle or more enriched in minicell. To enrich specific proteins in minicells, we came up with an idea of using polar localization signals. By fusing a target protein with these signals, it can be directed to the cell pores, which are the site where minicells are generally produced. So the protein becomes enriched in minicell. In bacteria, several polar localization molecules have been identified, and these include TSR chemotaxis-associated vector, PTSI from phosphortransfer system, some RNAs such as ARGZ and RPOS, and the PopG protein, which is involved in cell division of Caulobacter, but also localized to pores in other bacteria. When we fused a reporter to these candidates and tested their accumulation in minicells, we observed no increase in enrichment when mRNA molecules were used. But when polar localization proteins such as PTSI or TSR, when we employ these proteins, the enrichment level in minicell increased by about 50% compared to the control. Also, although the molecular mechanism behind is not fully understood, when we used a heterogous polar localization signal as PopG, the enrichment level was even greater, up to about 20-fold higher. I believe using this signal protein could be very helpful for further engineering and programming of minicell for particular application and programming. So to wrap up my talk, by manipulating cellular economics and physical constraints, we can better allocate resources and engineer bacteria effectively. And this approach could be a powerful approaches in synthetic biology and biotechnology. Well, thank you for your attention, and I'd like to thank my lab members and collaborators for their support. Thank you. That's all I prepared today. I hope you enjoyed.
Fantastic talk. I love it. Actually, many of your work also sort of, overlap with my earlier work as a professor. Let me check with Blake whether he's still here. Okay, let me start with one question. Your
Sure,
first part of the talk that
sure.
basically link with the resource, allocation. There are
Mm-hmm.
two type of product you could make. One is a primary, product, and the secondary product is the other one. And
Mm-hmm.
then you mentioned ribosome, basically availability or resources is kind of, high when
Mm-hmm.
the cell grow very fast. So could you comment on which product is more effective, primary product or secondary metabolite?
You mean by the ribosomes, you mean?
Yeah, resource, availability, because I mean, the fast growing cell means, I mean,
Uh-huh.
that's mostly related to primary metabolite, but the
Okay.
secondary metabolite produced after, that fast growth phase, that's basically the stationary phase.
Mm-hmm.
So I'm not sure anyone to study on impact of resource allocation between, I mean, the effect of the cellular resources on the production of the secondary metabolite versus primary metabolite.
Well, to be honest with you, I have no idea about it, but at least in my approaches, I maintain the cell in steady state
Uh-huh.
of exponential page cell. Uh, when I use this chemostat, for instance,
Hmm.
where
Yeah,
is
you
that?
use the chemostat to
Yeah,
study,
okay.
okay, to maintain the everything in the sort of kind of exponential phase, you know.
Yeah, exactly.
So
So, be,
my question is, anyone kind of compare
mhm.
um basically impact between this kind of high growth stage versus stationary phase, because I mean the most of the secondary metabolite produced in the stationary phase, the behavior might be very different. That might be another important thing, because sometimes
Mhm.
secondary metabolite field, basically they kind of expecting those kind of thing happening in the later phase, and then primary metabolite basically produced during this kind of interesting high growth phase. So comparing that one might be good, I think, area
Okay.
of research in the future. That's why I'm asking.
Yeah, that's a really good point. To be honest with you, I did not think of, or we did not take into account that options.
Hmm.
But I think, yeah, you are right. We need to think of the metabolite in different growth pace. Even if it is growing very, very fast,
Hmm.
probably the production of metabolite can be different as
Hmm.
different cellular page. I need to check, and then maybe I need to come back to you later.
Sure.
Yeah, not to give a clear answer.
Yeah. So because I mean metabolic burden is very old, idea. I mean even 20 or 30 years ago, I mean even people
Right.
didn't know about metabolic burden, and then that's
Mm.
always the problem. But now we start to understand systematically or
Mm-hmm.
mathematically, using those kind of equation, those kind of Lebourgeois relation, etc. And then,
Right.
but still very complex problem.
That's true. Yeah, that's why many people are trying to reduce the metabolic burden, and this manipulating resource allocation is one of the approaches. Probably the other approaches can be just to uh, get rid of unnecessary genes, and then the cellular proteome size can be increased.
Mm-hmm.
Yeah.
I see the Blake just, show his face. Blake, do you have any comment or question?
No, I was really curious about what is the role. So, what studies have you done on scaling it up and seeing if you retain the engineered motifs that you've had? So, you go to great lengths to balance the cells, you go to great lengths to do the engineering of them. But there's a great saying in industrial biotechnology. Well, there are several, but one that I really love is something funny happened on the way to the fermenter. And things that work really well, like at these mini chemostats, when you take it up to 310,000 liter, things go amiss. So have you looked at anything like that, like predicting scale up properties of these cells at all? Or is that something, off the scale?
Wow, today's question is really, really challenging. Well, I already had a chat with my former colleagues, and we did not have clear answer regarding the scaling up because we didn't test that. But at least for this study, we also uh, used, and I also got some help from the computational approach to predict the gene expression activity and resource allocation behavior. But according to the computational approach, the prediction was accordant with my uh, experimental result, which means that if we get some parameters for large scale of the bacterial gene expression behavior, probably we can predict how much outcome would be produced in the cell. Well,
Yep.
just
Okay.
shortly, well, I don't have any clear answer, but at least we can predict using the AI-based computational tools.
Yep. And so the other question I have is, what if you had, so the idea of microbiome engineering, or synthetic communities to, be a core component of biomanufacturing moving forward. Have you looked at taking the same genus, the same strain, but with two different engineered pathways and putting them, populations of those cells together and then
Uh-huh.
seeing how they interact and see, things like competition, things like intracellular trafficking, things like metabolite sensitivity. I mean, to me, that's where you get into some really interesting
Hmm.
challenges in predictive science and metabolic engineering. And have you thought about that and what you would do if given that challenge?
You mean to make the better, I mean to grow faster and to produce more.
Well, maybe even synergies where the cells could help each other out. They're performing
Mm.
different functions, right? In addition to their innate core metabolism, right? But if Chris Voigt, for instance, had a great technology back in 2009, 2010, back when he was still at UCSF,
Mm.
where he had, I think it was the Helix technology, where he had one cell produce a product that was then uptake by a cell in the same culture to take it the rest of the way. That kind of handoff and partnering can happen within it. But I'm talking even more rudimentary than that, just cells that are different specialists, right? Different populations being mixed together and seeing
Yeah.
what the interplay is.
Yeah, I know what you mean. Well, actually,
Yeah.
I did not think about that option, but very recently I found one paper regarding biolacine synthesis. According to that paper, they made the synthetic consortium. Instead of putting the all biolacine synthetic genes in one cell, they split split the genes into different population. Eventually, by combining different cell population eventually more pyrrolysine could be accumulated in the cell. Maybe this is the example of what you are
Yeah.
thinking, I
Yeah, it
guess.
is. It is.
Yeah. So yeah, we can design the consortium, and but again, it is necessary to design very, very carefully.
Yeah, it's interesting in the synthetic biology community that there is a prevailing wisdom that you need to come up with the superhero bug, right?
Right.
That you need to have Superman, Supergirl, super eukaryote, super archaea, super, whatever. But I'm a big fan of the Avengers and the Justice League, and so I'm a really big fan of making a team of different unique individuals coming together and really doing great things together. So I think that's where we're headed, but we're a long ways away from that. But anyway, thank you for that input.
So Blake,
Yeah.
I'm very satisfied with your question because that is my direction in the future. I'm
Mhm.
following microbiome engineering. Actually, we have the big paper under review, and
Mhm.
then you
Uh,
just described exactly same way, specialists doing their job, but together we see huge synergy.
Yep.
And hopefully that paper come out this year, and then I will probably introduce that paper to you.
And then I'll bring you back to JBay to give a talk about it. That'll be great.
Okay, wonderful. Thank you. Thank you so much. That's wonderful. So yeah, I do believe, I mean, we used to think about, Superbug, but Superbug cannot do everything. But
mhm.
now, I'm thinking about why we talking about only one bacterium, one feedstock, one product in one bioreactor doing everything. I do
Mhm.
believe we can make, microbiome or even just simple synthetic consortium that kind of doing amazing job because that's what nature do. I mean, nature already have multiple huge amount of, number of the bacteria doing something together. And then I believe Adam Arkin in your side basically also doing amazing job, even computationally, and then using synthetic consortium doing something. But actually, I'm try to translate that idea into real world product generation. That's what I'm doing right now.
Well, even in nature's harshest environments, you still have communities that thrive there.
Mhm.
And I'm coining it multiplexed biomanufacturing,
Right,
where you take biological diversity as a way to take all comers and turn it all into products. If we're ever going to beat refineries,
right,
we have to be like them from
right.
an industrial state of the art. We have to be able to take multiple inputs and convert everything we generate into a value added product and waste not, want not, right? We can't be the one and done
Right.
shop in terms of competing with chemical engineering, right? It has
Absolutely.
to be more like what they are
Right.
and for it to scale and be affordable and profitable.
Yeah. And to give us teaser, if we use one bacterium, for example, in the same, mixed, substrate condition, that's actually toxic to the bacteria.
Mhm.
One bacterium cannot, consume more than, for example, I don't know exact number, but 70% of the entire, substrate. The other was also same. But if we combine two things together as a consortium, we, seeing, almost 100% consumption of these toxic,
Yep.
chemicals in the mixture.
Yep.
So,
We're not even talking about the roles between prokaryotes, eukaryotes, filamentous fungi,
mhm,
you
mhm.
know, and even like lichen out there in the world that are fascinating microbiome to study that could really open a whole new realm of possibilities for us and in biomanufacturing.
Absolutely. And also, Juhyun also, also talking about some computational analysis. Actually, we do actually, but this is very, interesting, complex kind of computational challenges. And I believe, people like us, together, we can solve the problem together. Absolutely.
If you could predict what syncom you need to build in order
Uh-huh.
to take all the carbon and push it all the way to the product or multiple products, that would be huge.
Yes,
So,
that's it.
yeah, I absolutely agree with you.
Yeah, that is actually my, current project we are working on for multiple years, and then we have to have the final first, outcome out of that, many years of effort.
I will follow up with you very shortly.
Yeah.
Absolutely. Wonderful. Any question or any comment? If not, actually I see the o'clock, and then now 1 a.m. Korean time, Juyeon's time. So
I need
we
to go to
close.
bed. Yeah.
Yeah,
Thank you.
okay.
True, thank
So
you so much. True, thank you so much for staying up. Go have an adult beverage and go to sleep. Yeah.
It's my
okay, let
pleasure.
me
I'd like to
close.
see you in person.
Uh-huh. Let me close by saying something. Okay, thank you all for joining us and staying here. We will meet again on August 28th, Thursday, the same time, the same Zoom link. We'll have Dr. Lead, Nasser at National Science Foundation, Emerging Technologies, Biotechnology, or TIP Directorate, that's a new directorate at NSF, and also Silly Ong Lee at Rice University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thank you, and I stop recording. Just 1 second.